Alright guys, this video is on cells. So this really covers a pretty broad spectrum of concepts within the cells. So we'll go ahead and jump right into it. So it's broad, but not terribly specific. Right, so we always start off with a bunch of questions and then try to look at these questions again when we are done with the video. And hopefully at that point you can answer all the questions for yourself, which is important to be testing yourself as opposed to just listening to somebody talk in a video. A lot of times the information doesn't stick unless we're asking ourselves questions. So let's see, defining feature eukaryotic versus prokaryotic cells. What regulates which molecules allowed to enter and exit the cell, ribosomes, what do they do, smooth and rough ER, what are the power generators, why do we call it that? Okay, so those are all really good questions, right, that you could easily see on the exam. So let's go through all this stuff, okay? So we start off right here where we just have an image where we can see a lot of stuff that we're about to go over, such as the nucleus, the plasma membrane, ribosomes, smooth and rough ER, Golgi apparatus, mitochondria, lysosomes, and cilia and flagella. All that stuff we are going to go over here. All right, so let's go ahead and jump right in. So we start with cell theory. So cell theory states that all organisms are made up of at least one cell. Cells are the basic unit of living things, and all cells come from pre-existing cells, right? So you could imagine a multiple choice question about cell theory, which says like which of these concepts is not part of cell theory, and they list all three of these which are true, and then one that is untrue. Cell theory does not say that cells are made of multiple cells, but they are made of at least one cell. So that could be an example of an incorrect multiple choice answer. It could easily show up. All right, prokaryotes and eukaryotes. So eukaryotes have internal membranes while prokaryotes do not. A prokaryote is a unicellular organism that lacks a membrane-bound nucleus, mitochondria, or any other membrane-bound organelle. Prokaryotes are divided into two domains, Archaea and Bacteria. The first living organisms were prokaryotes. A eukaryote is any organism whose cells have a nucleus. Eukaryotes can be unicellular or multicellular. The defining feature that sets eukaryotic cells apart from prokaryotic cells is that they have a membrane-bound organelle, especially the nucleus. Eukaryotes evolved about 1 billion years ago, while prokaryotes evolved 3.6 billion years ago, right? And so basically, one question, if any, on prokaryote, eukaryote, and you just want to have what is the defining difference between the two, right? And again, being that eukaryotes have membrane-bound organelles, that's the part we really want. Okay, the nucleus. So the nucleus contains most of the cell's genetic material, which is organized as DNA molecules, combined with a large variety of proteins to form chromosomes. The genes within these chromosomes are the cell's nuclear genome and are structured in such a way as to promote cell function. The nucleus maintains the integrity of genes and controls the activities of the cell by regulating gene expression. All cells are surrounded by a plasma membrane. The membrane is composed of a phospholipid bilayer with proteins dispersed throughout the layer. The membrane is also covered in places with cholesterol molecules and proteins. The plasma membrane is selectively permeable and regulates which molecules are allowed to enter and exit the cell. Okay, so that's big information right there that we want to have. The surface area of the plasma membrane, which has control over what enters and leaves the cell, limits the overall size of the cell. The reason for this is that the volume, which is 4/3 pi r cubed, increases at a faster rate than surface area, which would be 4 pi r squared. Therefore, the cell volume increases to the cube of the linear increase, while the Cell surface area increases only to the square of the linear increase. Right? Again, don't worry too much about those details. Right? Surface area of the plasma membrane has control over what enters and leaves the cell. Right? That's important, and it limits the overall size. Alrighty. And before we jump into ribosomes, just a quick little part about the nucleolus, which is involved in ribosome assembly. The nucleolus is a substructure of the nucleus. It transcribes ribosomal RNA and combines ribosomal proteins to create the large and small ribosomal units. Okay, and then we jump into ribosomes. Okay, so ribosomes, we're thinking about protein synthesis. Ribosomes are both synthesized and produced in the nucleolus. The ribosome is the site of protein synthesis or translation. Ribosomes link amino acids together in the order specified by messenger RNA molecules. Ribosomes consist of two major components, which is the small ribosomal subunit, which reads the RNA, and the large subunit, which joins amino acids to form a polypeptide chain. Each subunit is composed of one or more ribosomal RNA molecules and a variety of ribosomal proteins. Okay, looking at smooth ER, in which we're thinking about detoxification and lipid synthesis. Smooth ER is devoted almost exclusively to the manufacture of lipids, and in some cases to the metabolism of them and associated products. In liver cells, for example, smooth ER enables glycogen that is stored as granules on the external surface of smooth ER to be broken down to glucose. Smooth ER is also involved in the production of steroid hormones in the adrenal cortex and endocrine glands. Large amounts of smooth ER are found in liver cells, where one of its main functions is to detoxify products of natural metabolism, overloads of ethanol derived from excess alcoholic drinking, and barbiturates from drug overdose. To assist with detoxification, smooth ER can double its surface area within a few days, returning to its normal size when the assault has subsided. And then what about rough ER, right? Which has much more to do with protein translation. Rough ER gets its name because it is studded on its outer surface with ribosomes. These are called membrane-bound ribosomes and are firmly attached to the outer cytosolic side of the ER. Rough ER is found throughout the cell, but the density is higher near the nucleus and the Golgi apparatus, which we're going to talk about right now. Golgi apparatus. The Golgi apparatus receives proteins and lipids, which are fats, from the rough ER. It modifies some of them and sorts, concentrates, and packs them into sealed droplets, which are called vesicles. The Golgi apparatus is located near the nucleus and has several flattened membranous sacs. And then what about mitochondria and ATP? So mitochondria are rod-shaped organelles that can be considered the power generators of the cell, which I believe was one of our questions. If we jump all the way back to those questions. Where are you? What are the power generators of the cell, right? So we just started talking about that. That would be mitochondria. Okay, so power generators of the cell, and they convert oxygen and nutrients into adenosine triphosphate, or ATP. ATP is the chemical energy currency of the cell that powers the cell's metabolic activities. This process is called aerobic respiration and is the reason animals breathe oxygen. And you should know that chloroplasts produce ATP for photosynthesis in plants, while mitochondria produce ATP for aerobic cellular respiration in animals. Lysosomes are involved in waste breakdown. So they break down cellular waste products, fats, carbohydrates, proteins, and other macromolecules into simple compounds, which are then transferred back into the cytoplasm as new cell building materials. To accomplish the tasks associated with digestion, the lysosomes utilize about 40 different types of hydrolytic enzymes, all of which are manufactured in the ER and modified in the Golgi apparatus. And then finally, we have cilia and flagella. They move liquid past the surface of the cell. For single cells, such as sperm, this enables them to swim. They consist of a cylindrical arrangement of 9 microtubules. A pair of single microtubules run up through the center of the bundle, producing the 9+2 arrangement. And then finally, we want to know cilia are short and flagella are long. Okay, so as I said, we kind of really briefly go over a bunch of these, and we get back to our questions here at the end. So this is a great time to hit that pause button and see if you can answer these questions and just be able to describe, ranging from the nucleus to the plasma membrane to ribosomes to the ER to Golgi apparatus to mitochondria and ATP. Sentence or two describing what the role of each of these are. And if you know that, then you're pretty good to go for this section.